and we'll just do something regarding that. And I will just give you a broad eye view of where murburn concept is. And then today, we just, in the morning when we got up, I saw biological chemistry had rejected our paper on cyclooxygenase. Wonderful rejection letter. Smooth, sweet, no nonsense, very cute. So I wanted to expose you to all aspects of research. So that will come later. Danny will, I'll quickly in about 10 minutes, I'll cover some aspects that I missed out regarding electrophysiology yesterday. So now, very fundamental aspect is that now you see what is written here. Can you see what is written? The first few italicized papers are about the foundations of the experimental foundations that led to murburn concept, and the remaining are the applications of murburn concept in diverse fields. So that's what the papers are in decent journals. So this thing, if you have, you can always follow what murburn concept is. This is solid. I do not need do not need any further endorsement in respect to publication and all that. Publish, done. Now, but it can get rooting in society only if you people do something about it, because otherwise it's not going to happen. So with that, let's just quickly get over the remaining slides and then go to Danny's content, and we will break at around 12:30 for Professor K.V. Venkatesh's talk. He has assured me that he'll come. He's swarmed in meetings, but he'll come. 12:30, he's now in Australia, so it will be evening for him. So otherwise he had promised to come here. So now this is what regarding this guy, pumping and binding logic. I told you that this is the protein. So now you had these molecules, that steroidal molecules. They were supposed to, do you remember the structures of those molecules? And now you see here, the kind of inhibitors that they are, modulators that they are for NK. You see, it goes all over the ballpark. All over the ballpark. Okay? Steroidal, non-steroidal, some molecules and all that. Now, the point is, all these are redox active, interfacial redox active. But sodium potassium ATPase is not a redox enzyme, right? It doesn't have any cofactor. It is just hydrolyzing ATP and pumping sodium potassium here and there. Now, how do all these affect it? These can all affect it because of the murburn theory. Murburn theory says that it is based on oxygen's redox activity, which moves the potassium in and sodium out because of these things. This is the reason why we have that sieving. You see, potassium, if potassium will be inside in the presence of electrons, it will be minus 19 plus 3. Inside, sodium will not be that preferred because it is plus 2.1. Did you understand that? No? Yeah, you understood that, no? It is the thermodynamic reason. It's not because of some willful pumping. Do you understand? No enzyme can do that. Okay, so then I wanted to show you here miscellaneous. It can explain simple structure and oxygen sensitivity, uncouplers, how they affect observation of ATP synthesis by NKA. So what they did is, there was Freeman Cope and John Glynn. John Glynn was a Cambridge biochemist. He won. He said ATP is synthesized by NKA, reverse of ATP hydrolysis. But then the synthesis is because recruits ROS. Do you understand? So misinterpretation. Then the transdermal peptide. I told you something, that structure you see, lysine, histidine. Okay. So this kind of, I told you in cytochrome P450 that N term had lysine, and that messes with the ROS dynamics. So then these diverse molecules, and see, The phosphorylation aspect. This phosphorylation is seen on so many places here. There is a phosphorylation here also. How can the protein get phosphorylated there? That is a transmembrane region. If it is by ROS, it can. Not by an enzyme. Did you understand? Did you understand? So the phosphorylation is not just seen. What they said is that, see, the binding of ATP, then the binding, it gets phosphorylated. So they said when it gets phosphorylated, it has got affinity for potassium. When it is non-phosphorylated, it has got affinity for sodium. How? Mazak hai? It doesn't happen like that. The thing is, the phosphorylation is not just occurring at one place, multiple places. That is a stochastic phenomenon, because once ROS is there, it can transfer a phosphate to any OH group. So this ADP has OH group, that's why phosphate gets attached. Serine, threonine, all those also have hydroxyl group, that's why phosphate gets attached. No enzyme can go into this lipid filled region, but a small radical can go. These are, kind of supporting evidences, for the new theory. So I also, you see the assessment of, if this I had shown you, one gate opens, then potassium goes in, then closes, then something goes out, then that gate open closes. Even if you basically, for non-selective and reversible process, the overall possibility of this series is 1 by 800,000, whatever, the huge number. Even if you have a selective and higher binding value of half, the overall score would be only 1 by 16,000. Because everything is reversible, because the same molecule is doing the process. So that is what I have shown here. And what the questions at the outset. Okay, no reason provided why ion permeability changes during various phases of action potential. Why? Why should they change? Potential change, the protein gets permeable different. The protein makes the potential. It doesn't make any sense. So many writings, this Farachi's writings, Tasaki's, Terakawa, all these people's works were completely sidelined, cherry picking data, not making global sense. So this is something. And the pumping thing, you get the cellular in to out pumping and out to in pumping. If you just put your common sense to it, you will see that it doesn't work, because all that this 3:2 achieves is, one, sodium getting pumped outside extra, right? Now what will happen if it constantly pumps like that? All the sodium from inside will get pumped out. And what they say is, no, there are also channels to allow them to go back and forth. So it is like making a picture for you which does not explain anything. One guy goes to a shop and he buys, he takes some 10-12 banana. Look at me, this is more important. 10-12 banana, one gucha mein he takes, and he says, bhai ye kitne ka diye? How much for this? He says, 30 bucks. So he says, okay. With the other hand, he takes 2 oranges. Ye kitne ka? How much for this? Roughly around 30 bucks. Okay, he keeps the banana down now. He takes an apple. He says, how much for this? One apple is around 30 bucks. Acha, then he puts the orange down. Okay, then starts biting the apple. Apple ka paisa dijiye 30. I put orange. Orange was in lieu of that. So orange maine liya nahi. I didn't get the orange from you. Or he says at the banana, orange, I put banana ka paisa. Banana maine liya kahan? Where did I take the banana? There is no real accounting. It is just pushing one thing to there. When you ask, like the mirage that MC Escher drew. Did you understand? At one point, one thing substituted for the other, but on a global scale, nothing explained. So that's what this, if you also see the dimension. So for one cell, if a macrophage changes the dimension, the surface volume ratio and all change. How does the system account for that? How does? There is no logic to account for that. You understand? Suppose a cell is there. Have you seen a macrophage change its shape? It can change volume to surface area very dynamically. How can it maintain its concentration when it doesn't make sense? So these are the kind of things that I wanted to show you. And I also wanted to say that murburn concept is also very important for diverse protein modifications. The hemoglobin glycation and all that, it's because of murburn activity only. All right, it's a redox process. So basically, like murburn modifications, you can also see in non-accessible areas. So this is the elaborate scheme of murburn post-translation modification. Okay, so the zeitgeist is science advances by negation, society served by correction. Uh, uh, we have to have multiple aspects, global data points, thermodynamics, kinetics, structure, function, all that must appeal. Only then it becomes a theory. So with that, I give the stage to Danny. Yeah, around 12:30. Aidcha. Yeah, 15 minutes is enough. Cyclooxygenases, do it. Yeah, you can do that. Come. Why are you not doing? Professor, we are waiting for Professor Venkatesh. So.
Yes. What are the energetics involved in modifications of a? Is there a unit loss which will create this much of conformational change? Can that energetics be derived?
No, we have not done anything like that. What we have done at this moment is, Prasad.
It is more of observation.
Yes,
And
rational, qualitative reasoning.
Qualitative rational explanation has been given.
Yes, basically, now we are observing so many things, right? Now, to all the phosphorylation, all the glycations, and all that, um, there are so many possibilities that can happen. Only an enzyme-mediated phosphorylation possible. A ROS-mediated phosphorylation also possible. Similarly, glycation, it is also possible. So sometimes when what biologists generally do is protein structure, when they do not understand certain things, when they do not have direct tangible explanations, they always resort to what? And lo and behold, conformation change occurs. That's a very sometimes very relevant explanation. And then affinity changes. These are the two reasons. But then why should a conformation change occur? Why should an affinity change occur? These are important questions. Prasad asked me, what is the energetics, in the sense that have you followed the exact sequence or accounted for something went in?
My I am from a structural biology.
Yeah.
I believe in that, and some structures have to happen, and the structure provides the infrastructure or the basic mechanical support system for the chemistry to happen, where ROS may come in. I am very happy for that. Okay, but your ROS system is already happening. But to enable that ROS system to work, or murburn concept to work, my question is that how much of this ROS to conformational changes the energetics are related. So what I mean to say is, there is an enzyme, suppose a kinase is there. A kinase, one flip happens, and that flip makes one active to inactive. And in this, suppose, for example, hypothetically, a ROS is involved in that. So how much ROS is needed for how much of conformational?
That depends. So that depends, you would imagine, on where the
unit of and because if ROS is playing a role in ATPase movement, because my what is that called, excitement in ATPase molecule is, I'm coming from an engineering background. So when I see a motor and a stat and struts and all those things and the gear system, I get excited, and I have worked for past 25 years on that. Now, yes, that particular infrastructure might be there, a support system is there, and your ROS is also maybe complementing it that in some other way. But where is that energy coming from? And is it that completely we are disallowing the concept that there is no need of energy? It is just raw species which are coming and then going off.
No.
And so how do we? There is a
I get it.
structure.
I get it. So what you mean to say is your approach is from the perspective that structural information of the protein is what allows catalysis, right?
It is enabling catalysis.
Yes. Now here is something. Now the other aspect is the energetics part of it, the overall aspect.
Which has to support the whole theory.
Yes. Now the point is that now when you put oxygen into any system, even at room temperature, there is spontaneous formation of ROS with electron sources here. There is, so it's an experimentally ratified thing that there is effective charge separation occurring. Oxygen is able to receive electron. So it is, even in room temperature, it is a facile process, and that chemistry of electron transfers of this is well established. So the feasibility or the energetics is good. Now, how much exactly ROS is required to denature or transform or protein?
Yes.
That depends upon each specific protein, I would imagine. So now if you have a protein that has a very crucial amino acid.
Sir, simple question. Simple. In your own analogy, I am traveling from here to there. The velocity is 2 meters per second. I am not traveling 2 meters, but per second I am traveling 2 meters, right? So similarly, how many?
No, that's what I address. I understood that question. That is what I'm addressing. Now, if you have a protein like this, you want some change, structural change to happen in this protein. And if you believe that ROS mediated phosphorylation or some hydroxylation or oxidation, something that could happen, what is the energy term that is required? How much ROS would correlate to a protein getting modified or changed? Is that?
I don't have that vocabulary for that. How many ROS molecules will give me 1 angstrom or 0.5 angstroms of conformational change?
I understood that question. Now, the point is, now, suppose if I do this, अरे यार गुदगुदी तो होना चाहिए तेरे को मैं example का बेड़ा गर्क कर दिया. So you touch him in the right spot, he'll react, right? So that's what I'm saying. Now the protein has a particular conformation. It is energy minimized with respect to the surroundings. Now that if you can perturb at a particular place, it will get affected more in response to some other thing, or
say there are hotspots.
Yeah, there are hotspots.
Yes.
So now the point is, your question is how much of ROS, or what is the total energy term for n number of ROS to affect something tangible that can give some kind of structural change or something like that. It depends upon protein to protein. It depends on ROS. It depends upon environment. It will depend on that. That's it. That's it. It has to be. It has to be.
Planck's constant is there.
Yes.
It is universal.
No.
Pi is there. It is universal. Similarly, for these many ROS, or per single ROS, this is the change.
No, you did not understand one thing about ROS. ROS, how many ROS are there? Yeah. Superoxide, singlet, hydroxyl, peroxide, all these. Yeah. So now and then
are you able to, in your mechanism, explanation of mechanisms based on murburn theory, okay, are you able to identify which ROS is actually responsible for that particular? Most likely, yes.
And I can't really, because when you have superoxide, they dismutate spontaneously forming peroxide. And then peroxide and superoxide can react. You get hydroxyl radical. And they're all in interactive equilibrium with each other. And the question, I think your question is from a physical chemist perspective, or more the structural engineer's perspective of a protein.
Exactly.
Yes.
I would like to, because I have seen things happening in that way. You are telling about big molecules here, I mean nanomachines, which I teach them as nanomachines. Okay. So you are talking about nanomachines, but what I would like to, I have proved in a patent draft is that even engineering a particular simple monomer like aspartic protease 325 amino acids, it has various layers of machine components. So I identified them and how they relatively move to each other. That is creating the function. Okay, there in this particular scenario, most likely I don't know whether ROS is involved or not. So there may be a boundary where ROS is effectively acting, and that is the explanation. Or if ROS is playing a role in this also, how that particular, I told you that 7 angstrom are the lysines or the charged polar molecule amino acids in a particular protein are able to create this ROS spontaneously and result in that particular function to happen. So this particular concept. So my very purpose of coming here is there a way I can further apply or further contribute to this murburn concept, and that's the main thing which I'm interested in.
Yes, I want to show you some things like now a protein is stabilized, energy minimized with respect to say there's a serine here, there's a glutamate here, it enters a hydrogen bond there, something like that, something it is like that. Now comes a raw species, attaches a glycate molecule or a phosphate molecule or something. That interaction is perturbed.
Yes.
Now it depends upon where the protein is, what kind of protein it is, which part of the protein has been randomly modified. That's what I'm saying.
So in that case,
so because we are not controlling where the ROS goes, but now the future of murburn is, now we saw the sodium potassium ATPase, right? The protein will have a ROS and ion dynamics through it or near it. That will be a probabilistic route. Now those probabilistic routes, if they are altered, if they are modified, if they are messed up by those diverse kinds of molecules that are shown, then you will have impact. So it is now, I keep saying this, there's a mountain there, if you put some water from the top, it will come and get accumulated in a pond or some other place at a lesser height. It will follow multiple routes. This is the whole point. When ROS reactive species, diffusible reactive species is involved, we have always looked at cellular processes with only the structural aspect of the protein. Here what we are looking at is something that can affect the structural aspect, but there is an interactive dynamics of molecules, these ions and the reactive species that can also affect the overall outcome. The protein affects something on its own, then this interaction of these things outside the protein or around the protein can also affect. And the protein can also get affected. That's what I showed in this post-translational modifications.
So I mean, there is a chance that there is a skeleton. As much as you are passionate about ROS, okay, and nature's simplest principles, nature also has its own simplest way of doing things with respect to the creation of a skeleton for a particular mechanism.
Yes.
So in that case, where is ROS play in collaboratively working or symbiotically working with the structures? So that is what my intent.
Yeah, I understood. The point is that ROS dynamics is what allowed the evolution of proteins. Because, or ROS, no, diffusible reactive species. Because fundamentally, they are the ones that are energetically driving things. Because if you see the cellular fundamental energy pathways, 2-electron processes, they are always around the glycolysis and all that. They delta G 20 kilojoules here and there.
How much of light from external source can influence the cellular internal mitochondrial cell walls in me?
Well, I was talking about the photodynamic therapy yesterday. There, when you
this light is influencing,
yeah,
and that means this light has the ability.
And I also talked about how one mitochondrion kept here.
Correct.
Yeah, because it's a high energy reaction. That's why.
So
that's what ultimately.
The whole macroscopic energy is influencing the nanoscopic or picoscopic.
Sure. Regimes.
Creating oxygen. This redox. ROS.
Yes.
It is creating. So that means any disease, can I control by this type of external? I am thinking.
Wonderful. That is our paper we had sent to Cell Reports. Exactly, that's what I'm saying. Now here, Lauren Jekin and I, we had put down a paper wherein this is a system, interactive system, wherein it can generate radiations and it can use radiations. So there are cancer cells that lose their tumorogenicity by changing its location, the kind of the contacts it has the kind of radiation it gets, all that. So matter can affect, can produce radiation, radiation can interact with matter. In fact, that particular thing is being shown in the last talk that Laurent will give. That's the final talk. So basically your question that how do the energy term interact with structural aspects, right? That is what you are coming to, right?
In a way, but there is a lot of synergistic aspect also I am expecting, just not disregarding the nature's structural build it has done all the...
No, I'm not disregarding it at all. I'm saying I'm complementing it. I'm never ever no, I clearly stated that in multiple occasions. We are not negating just because I've discredited certain affinity based theories in certain models. I'm not saying that there are active site, there are no active site, there are no structural effects.
You are understanding that particular aspect very clearly. So why don't this aspect be synergistically explained about this bacteriophage, about this ATPase molecule, not and about the flagellar?
Oh, sorry, I did not explain the ATPase molecule. Yeah, we have a paper on that. Yeah, so bacteriophage, I have no clue what you're talking about. What I want you to, I think, Prasad, what is happening here is we are not saying there is no structure based outcomes. Of course, very well established it is. What we are showing is a completely new interactive dynamics that we have overlooked. That's all. And that can also affect structure, that can also produce radiation, that can also, there's a huge window of opportunity to understand that. That's all. I'm not negating anything, saying that there's no movement and all, but then without movement also we can explain. That's what I'm saying. So sometimes certain things are not understood. People invoke only conformation change and affinity change. There are things which are interactive dynamics also can explain. That's all. So one second, Professor, has he come? Vandana, has he come? Has he come? Has not come. So Danny, you start. If he comes, yeah. 1 second, we are not. We'll take the interaction later. We have enough time. All right. Yeah. So Danny, you go ahead and speak, Danny. We'll take it because Danny's lecture is prepared. So this is contextually very important because we worked on this enzyme. Have you heard cyclooxygenase? An enzyme called cyclooxygenase? No?
Yeah.
You have COX. It's called COX. COX1, COX2. We have kind of studied the whole mouse, human, COX1, COX2, all that. So you will not be happy certain things that we say now.
No, I'm here to.
Yes. Yes, that's what. We are also complimenting only. That's good.
Okay. Like Sir said, these cyclooxygenases, perhaps one of the most important enzymes in the drug industry. Because you see that there are certain omega-6 polyunsaturated fatty acids like arachidonic acid. This is the primary substrate of cyclooxygenase 1 and 2. Cyclooxygenase 1 is known to be present in all tissues and it has got some functions, right? Some homeostatic functions. And it's kind of expressed all the time, you can say. It's there for some functions in the cells. But cyclooxygenase 2, COX2, is inducible. It's an enzyme that can be produced when it is necessary. So you can see that arachidonic acid is metabolized by this enzyme. This enzyme is like the Roman god Janus, as we have said, two faces. It has got two active sites. One is a more lipid buried active site. Next slide. Okay, there is a more lipid buried active site which is oriented towards the membrane, where oxygen is known to go in, and two molecules of oxygen are utilized per fatty acid. AA here represents your arachidonic acid, whereas the other side, diametrically opposite almost, you got another cavity which is very large. It's like a cave, I would say. There is heme right there. We have worked on so many peroxidase systems. We very clearly know that murburn reactions occur in peroxidase systems. Now we just needed to translate that into the cyclooxygenase system, because what we have here is the active site is buried, arachidonic acid is liberated by phospholipases, it just has to go in and then it has to get converted and come out. But there are some fundamental questions that we want to ask. So all of these, what you see here is that the ion goes through different redox states, and then there is a generation of something called a tyrosine radical. I want to keep it very short. This tyrosyl radical, especially tyrosine 385, is generated. And that is the agent which drives the catalysis, they say. That if you go back to the previous slide, we can see that there is a carbon 13 where attack happens, and it converts into, see, what you have is like a linear molecule. Of course, there will be some kinking because of the double bonds, but it's like a thread. It goes in like a thread, but it comes out more like a hairpin, right? So you got PGG2, which is the product which is formed from cyclooxygenase site. There are two sites, cyclooxygenase site and peroxidase site. So the next product is this PGG2, which is generated from the cyclooxygenase site, gets liberated, goes all the way around according to the conventional mechanism, and it enters the peroxidase site and goes and binds there. And it abstracts 2 electrons from the heme center, which is there in the peroxidase site. And then PGH2 is the product which is generated, and downstream there are other tissue specific enzymes which make products such as other prostaglandins as well as thromboxanes. So prostaglandins are mainly the pain hormones which you have, and thromboxanes are involved, for example, in platelet activation. And so prostacyclin kind of does a function where it can prevent clotting and things like that. It's a good thing, but there are bad, mostly those other hormones which are generated. Now we go to the next slide after this. So if you see the structure of both cyclooxygenase 1 and 2, if you see the active site, if you see in this A diagram, the green thing right there in the center, I don't know whether you'll be able to make it out. In the crystal structure of a particular rat cyclooxygenase, arachidonic acid is bound there. So you can see that all these amino acids, they say, are involved. There is some kind of van der Waals force that is able to accommodate all that. And there is a particular arginine 120 at the active site mouth, which will stabilize the COO minus part of the arachidonic acid. So what we can question is that the rate of oxidation of arachidonic acid is far better than many other polyunsaturated fatty acids. If it's the same kind of mechanism where a particular active tyrosyl radical is involved, why would that occur? Why should there be a disparity in the oxidation rates of various fatty acids, which occupy pretty much the same site? Okay, and this molecule, arachidonic acid, has to adopt a kind of L-shaped conformation, Now, I don't know, sir is here who is very good in dynamics. He might be able to tell me how a fatty acid molecule, which is so long, would go like 25-30 angstroms in. What would be the driving force for that? How would it come out like a hairpin? So these are fundamental questions which I had. So you can see that part of it is buried monotopically associated with ER membrane, major place where cyclooxygenase acts as the ER membrane. There are other membranes also it can associate with. So the drug molecules are, in the liver microsomes, for example, in the ER, smooth endoplasmic reticulum, then the drug has to enter in through the cyclooxygenase site, and the drug, that you have, many inhibitors, we are taking aspirin, we are taking in so many other inhibitors. How do those inhibitors act? And there is a lot of literature which says that ROS are involved. There are several things like hydrogen peroxide and singlet oxygen and superoxide. All these are also there in the cyclooxygenase system. They have been detected by spectroscopy. What is the role of those things there? Are they wasteful, useless products of aerobic respiration, as conventional literature says? So there are minuscule differences. They say that cyclooxygenase 1 has got a smaller pocket, cyclooxygenase 2 has got a slightly larger pocket.
Hello. Can you put Professor Venkatesh's video on? Is he on? Sir, can you come? Can you give me a video? Yes. Wonderful.
Yeah. Hello.
Hello, Professor Venkatesh. This is my audience here. Very happy to introduce. Please put his full face here. Yeah. அவரோட full face போடுங்களேன். Yes. Professor Venkatesh is a chair professor and is a chemical engineer. He's a systems biologist. He's a specialist of the field that we are doing. I had the good fortune of being friends with Professor Venkatesh junior and fellow colleague Sharad Bhartiya. We had had studied in this IIT Madras. We were in the same hostel, and something that impacted me terribly was my friend's death, and Sharad Bhartiya's friend was also he. So Professor Venkatesh is an expert, and he has Medflux. He has started this entrepreneurial initiative, and he has 24-25 people. Am I right, sir? You have 24-25 people with you. You're working, and he's a very senior professor and very respected, very high-profile professor from IIT Bombay. And I've been very fortunate. He was the convener of the first workshop on murburn concept that we conducted at IIT Bombay. Now I would like to leave the stage to him, and kindly, sir, please give us whatever you want to say. Thank you so much.
Thanks, Murli. I'm sorry, I was supposed to join the first day. You're able to hear? All of you are able to hear? All of you?
Please. Yes, we can hear you very loud and clear.
Okay. Yeah. So I was supposed to join the first day itself, and I've been traveling to Australia. So I got sort of caught up in the work here. So I'm just thought I will connect sometime so that I can meet up with you guys. So what I will do is I'll just take 15 minutes to tell you, the approach of systems biology and how that sort of connects to what Murli is trying to do in the Mormon concept, and how the energetics and thermodynamics come together, right? And then one of the approaches that one can take, irrespective of the hypothesis that you would want to put. So I can share it, and let me just share the screen. You're able to see this?
Yes, we are able to see your screen. Yes.
Yeah. So let me start with this video. So essentially, you're able to see the video, right?
Yes, very much.
Yeah.
Yes.
So this is a trigger, right? The square rectangle, what you have is a trigger, and you can see these cells, following these triggers, right? So it is optically activated, and as soon as that laser falls on it, it will start moving. Okay, so let me just show this again. So this is, a immune cell which moves towards a, say, for example, if you have a wound, right? So it has to go towards the wound. So so what has been done here is that you trigger a signal. So this is optically activated, and then once you have the signal, let me show you another video. Yeah, so you see the trigger comes here, then the cell immediately will start moving towards this signal, right? So that is how sensitive it is, and I change the signal, it will start moving to the other side, right? So how is this, how is this cell able to respond so quickly to a trigger, right? So you just put a trigger and then see it starts moving. So this looks like a engineering problem, actually, right? So although it is evolved naturally in the system, if you look at it, these triggers have actually come from, although a natural system, you can see that there is some design which is making it respond the way it is. Okay, so in a similar example that we have worked in our lab is also chemotaxis in E. coli. So how bacteria move towards a higher concentration of glucose, right? In this case, immune cells are trying to see where, based on a trigger, chemical trigger, it has to go towards a particular wound location, correct? So so for a given trigger, for example, bacteria in terms of a gradient of a glucose, right? In this case, the immune cell. So they are moving towards a particular signal, and then they respond to it. Okay. So if supposing a engineer were to design this, right? So they would, I can design this, with a problem statement that, I have to go up the gradient of a chemical, right? So that could be the problem statement. So can I design a system which will take you up the gradient of, say, glucose or a trigger? So if I have to do that, then I have to have a sensor. I need to measure the concentration of a trigger at a particular location, randomly move to another location, again measure, remember the old concentration, take the difference and look at that is a positive direction, right? And send that signal to a motor, right? And then it has to start moving in that particular direction. If it is facing in the opposite direction, it has to change the direction and then move in this direction, correct? So it's a typically what a physicist or engineer would design for a problem statement. Obviously, in this case, evolution has evolved these systems naturally. Okay, these are all swayambhu, right? So these systems have evolved by themselves to respond out in a Darwinian evolutionary pressure. They have evolved this for their own survival. Okay, so now we exactly know how E. coli is doing it or an immune cell is doing it, right? So you need a gradient inside the cell. You need a signal to be generated. You need those lamipodia and all that, to respond to the cell membrane to respond. Okay. Whereas in case of E. coli, you want the flagella to move in a particular direction. So now we know exactly how the sensing takes place. What is the signaling network? How that signal is transferred to a particular location on the membrane, and now the membrane responds either to a flagella or through other mechanisms for the motion to take place. Okay, so this is what I term as systems biology, where you are looking at a particular characteristic response, okay, of a biological systems, which may be characterized as a phenotype or a very specific characteristic response that you have to a particular input trigger. Okay, so you have input, you have a output, and then now the modern biology has given us all those components, okay, which connects these input to output. Once we understand this, then we have to look what are the design components which are present, how these feedbacks are responding, and then each one we should have a design equation, right? So this is how an engineer would look at and then see how the system operates. Okay, so in biological systems, just to set a context, right? So you want to look at right from genotype, transcriptome, proteome, metabolome, and then you have a phenotype, right? And these are hierarchically connection. So what I'm looking at is a external input going to the system. So this is a system definition with an output, but there are inputs which will trigger the system to make a gene, express a mRNA, which will express a protein, which will start a set of reactions or set a signal, and then the cell is going to, the system is going to respond in a particular way. So there is a hierarchical connection of this genotype to phenotype. And I give this example to students where, for example, you have a firefly, so you have a gene which gets decoded, and then you have a reaction which takes place, and then finally the firefly is going to glow, right? So if I transfer this complete connectivity to some other plant which was done in 1983, right? This is almost like 40 years back now, that you could transfer this complete phenotype to a new plant, right? So the tobacco plant also starts glowing because you have introduced the gene, you have made the RNA stable, you have made the protein, you have made the reaction to happen, and then you see that the characteristics has now been transferred. Okay, the response of this plant has also been now transferred because you have transferred this central dogma, right? So how do I link all these components and then see what is the design components that are present for a particular trigger and how the system is going to respond? What are the limits, right? Both the flux wise or thermodynamic limits on the system to operate. So these all become fundamental questions, just like an engineer would design a particular system to understand how it operates, right? It may be a heat exchanger, or it may be that I'm going to send a satellite to the moon, right? So these are all a particular input that I give, and then I design for a particular output to happen, right? So now we understand the physics in such a way that even I can send and make the satellite land on the moon, on the other side of the moon, on the dark side of the moon, which India could do it, right? So some in recent times, right? So if I ask the same question in a biological systems, obviously it is quite complex. And last 30-40 years, the modern biology has been looking at all these components and how they are connected, which is a massive amount of data that has been generated. So what has given us, your genetic, signaling, metabolic networks, and they're all connected in a particular way. There is a hierarchical link between cells, different cells coming together and then acting to create a complete system. And bioinformatics and other studies have also added to this knowledge base, right? And we have a complex system to understand and to see how a system is going to provide a particular output for a well-defined input, right? So what we have been doing in my lab is trying to quantify these systems with the input-output concept, trying to bring all the knowledge that exists, all the constraints that we know about, right from the capacity of the system to how they're connected, what are the feedbacks which are playing a role. So we bring all this data, including omics data, maybe single cell measurements or multiple cell measurement, tissue measurements. So we bring all those and put a constraint and try to see and apply the principles of system science, right, to understand how these systems operate. And that is what is systems biology. And that I've been looking at multiple systems of design component that exist, that the nature has evolved to give out a particular characteristic output. Okay, so just like an engineer would design something, then they will operate, then they would want to control in a particular operational space, and if there is any fault, we go and redesign it, and then we evolve, right? So so an engineer typically takes these steps of evaluating or coming up with a system, right? So you will bring components together and you design and you operate it for a particular characteristic output, and you want to control it, right? And then you create a if there are some faults or it's not working optimally, you would go back and then evolve it. Okay, so this becomes a continuous process of evolving a particular system. Biological systems also go through these process, but on a longer time scales, and the evolutionary pressures of survival of the fittest or some of those components evolve in a similar process, some of this design, right? And if for a particular fault or a particular perturbation, if the system is not able to evolve in the time duration that a particular perturbation exists, obviously there are chances that the system may get extinct also, like something like a dinosaurs that we have seen, that they don't exist now, although we know that they existed long time back, right? So how quickly are you able to evolve to take care of a particular perturbation, right? It comes in the process of that evolution, right? So I give this example of aeroplane, which is quite a complex system. So you have about more than thousands of computers which are trying to evaluate the pressure on the top, pressure on the bottom, keep it stable, right? Because the physics is, we know the physics of it. There are hundreds of feedback loops. There are millions of components. Okay, because this is a bottom-up design, that is, the humans have designed this from the bottom up. We have a design manual. I know exactly why I have placed a particular component into the system. Okay, now pretty much the takeoff requires manual effort. It's able to handle by itself. It's almost like an autonomous system that the flight is going to go from one place to the other, and it has to take off, be stable on the air, and then land at a given location, right? So that is its phenotype. That's its particular characteristic. Okay, and because it's a bottom-up design, we have a design manual available. Okay, but if you look at even a simple organism like Escherichia coli, E. coli, right? So the bacteria, which is one of the most well understood system, right? Although it's a single cell, it's quite complex. It's got about 4,400 genes. Even now, we don't know about all the genes, right? What is their role? There's a lot of connectivity between genes, mRNA, proteins, metabolites. So we said aeroplane has hundreds of feedback loop. A simple bacteria, obviously, you can look at it. It has got thousands of feedback loop. Okay, so it's an order of magnitude higher than even a complex aircraft that exists, that humans have designed. So this has thousands of feedback loops, and obviously this is a top-down system, right? We have not designed the system, right? The system already exists in nature, so there is no design principle available. I don't have a manual for E. coli, correct? And we think that E. coli, there is no competition. You see here, thousands of computers are being used to keep the system stable in the air. E. coli is now surviving in the environment, but it appears that there is no competition taking place, right? But obviously there's a lot of competition that is taking place, and making the cell decide, okay, what it needs to do, right? So although we feel that there is no competition, in every moment for its survival, there's a lot of calculation that is happening, and the system is in such a way designed that it is able to survive, or it is able to take decisions, right? So these are all happening through the control, the interactions, that all this connectivity that exists, the complexity that exists between the biomolecules is helping the cell take several decisions, right? So like the example that I was giving you, if there is a positive gradient of glucose, it will move towards the higher concentration, right? Because it's a food. So it will search and it will go towards the higher concentration of glucose. So that cell, that decision making, E. coli has that capacity because there is a design. So there's a glucose sensor, there is a signaling pathway, it has got all these components for it to move towards that positive. So a positive gradient of glucose. If I have a positive gradient of an antibiotic or a metal ion, right, which is toxic to it, it will run away, right? It is able to sense, and then it will say that you move the flagella in the anti-clockwise wide. You make the random movement of the body in such a way that it will start moving away, right? So it can move towards a positive gradient of a food, but it can also move in the opposite direction of a positive gradient if it is a toxic, like antibiotic or a toxic element, right? So that decision making it is able to do because there are design components inside, right? So now we know exactly how these systems operate, and how a input signal is translated, and how the system changes itself to move towards another direction. Another example is, for example, yeast, right? You put it on an osmotic pressure, right? So you put it under a high osmotic pressure, suddenly it will sense that, it will start producing glycerol inside the cell, and it will balance the osmotic pressure, and so that it is able to survive the osmotic pressure till it makes adjustment in terms of its a certain glycerol composition is built up inside the cell, and so the membrane is stable. Okay, till that time it'll stop cell division, it'll just survive. It will be in the lag phase, it will not do anything. Once it is the osmotic pressure is balanced, then it will start dividing again, right? So you will see the inherent intelligence that these systems have evolved through the evolutionary forces that in order to survive, in order to respond to the perturbations that from the environment, these designs have been evolved. So can I now build a manual for E. coli? Understanding. See, the plane had a very fixed phenotype, right? It was supposed to take off, be stable in the air, and land. And it is one of the safest way of travel now, because we understand the physics. We know even when a turbulence is ahead, the pilot indicates to you, right, that a turbulence may come, right? You put your belt, and sometimes they even avoid the turbulence. They'll take a roundabout route so that they don't hit the turbulence. So all this is possible because we understand the system well. We know the content. We know each component, why it is there, right? And so can I do something similar to these systems? Unlike a fixed phenotype, E. coli has thousands of phenotype, and depending on the environmental condition, it has to respond in different ways. Temperature stress, osmotic stress, right? There is a toxic element present, other organisms being present, right? A competition is happening with other organisms, so it has to respond and survive. So it has several phenotypes, and for that survivability, you need to have design components present inside the cell, right? The complexity that exists. So that's how can I now build if I know all the components, if I know the design principles, if I know how for each of those phenotypes the cell is responding, okay? And there are a lot of quantification numbers involved, just like in engineering system, right? It is just not that I have to bring two components together. I need to bring two components and then understand what is the operating values, right? So I can have a circuit, but I need to know what is the resistance, what is the capacitance, what is the operating constraints on these components, right? That I should understand. What is the binding constant of a protein, right, on the sensor, right? For example, glucose binds to the membrane. So what is its binding constant? All that plays a role in actual functioning of that particular system. So we need to understand, and it's almost like writing a manual now for E. coli, okay? For each of those phenotypes, given all the components, given the connectivity, right, and given the well-defined input, how the system is going to respond. So can I build a manual? So that is what is systems biology, trying to see how all these operate. Obviously, these components are complex because the input are many. There's a lot of feedback inside. Each component can interact in a different way, and then you have a different outputs that it has to be analyzed. So what is this complex system, right? So how do we even define this complex system? So let me just give you one example, right? So I'll just take another 10-15 minutes to tell you, and then I'll stop. So for example, I have a blue ball and a orange ball, okay? Now supposing if I randomly pick one of the balls, what is the probability that it's a blue ball? Obviously it is 50%, okay? But I'll slightly change the experiment. Say for example, if I pick a blue ball, in the next step I'm going to add another blue ball, okay? And then go on doing like this, right? So that is the experiment. The experiment is I'll randomly pick one, but I will not just stop there and then independently, mutually exclusively do this experiment. What I'm going to do is whatever color ball I pick, okay, I'm going to add the same color ball in the next step. So if I do this 50,000 times, right? So I will have 50,000 plus 2, right? 50,002 balls I will have at the end of the experiment. Now I ask you at the end of this, what is the probability that it is a blue or a red ball? Okay, I hope you understand the experiment that I'm doing. Okay, so
Yes.
if I do this, then obviously I cannot predict the probability, right? I always start with a 50% blue and red ball, but then I can have any probability, right? Going to a very small probability to a very high probability and even to the 50-50, right? So it all depends on the bias that I'm giving in the initial steps. Supposing I pick only the blue balls, right? Then the probability that I have a blue ball will be very high. If I go on putting red ball, then the probability of red ball will be very high. I can go alternative, blue, red, blue, red. So then I'll be 50%. Okay. So I can go to any of these steps. So this is what is the complexity. I've created one nonlinear step in the experiment. So what it has done is it has given multiple outcomes and it is dependent on history, right? So the outcome is dependent on history. So this is what is the basic concept of complexity, that it depends on the history, okay, and it can go to many states, okay, multiple states. So we all start as a single cell at conception, okay? So we are not very different, right? At that stage, only our mutations and SNPs that we carry from our forefathers is what is the difference that we carry okay, which may be less than 0.1%. So we all start as a almost similar system, but we all end up different, right? So now we are all not, we are all very unique outcomes that we have come out with, although the system was very, very much similar. Okay, that is because our own history is different, our steps that we carry is different, our own experiences when we are growing is different, right? So all this puts you in a different steady states, and we all become unique. Right? So this is a classic example of complexity, right? Which can put you in multiple states depending on the history that you take, okay, or the experience that you have had, that will take you to a new steady state, right? So this is the challenge, right? So although we say E. coli is one system, but each E. coli cell has its own journey that it takes, and it has a capacity to go to several outputs. So that is one of the reasons why bacteria also picks up antibiotic resistance, because there are some, okay, which escape this effect of antibiotics just because of the complexity that exists, right? So people did some experiments and they found out that something like 5 to 10 cells in 1,000 cells escape the antibiotic effect, and they get the chance now to mutate and then get resistance over time. So these kinds of complexities and the chance for evolution, right, happens only because of the complex system that exists, different components which have come together and then evolved with the evolutionary forces, these designs which have come. Okay, so just like engineering systems, we need to look at multiple steady states, initial condition, parametric sensitivity, the bifurcation, robustness, adaptability, evolvability. At some level you need to adapt to inputs, at some other level you need to come up with completely new design and evolve, right? And you need to be robust in some instances, right? In some instances you need to adapt. So these components are inherent in the system, and depending on how the perturbation occurs, the system evolves to give robustness, adaptability, evolvability, right? And then to evaluate this is what is systems biology. How do you put it in this perspective, right? So we look at nonlinear dynamics, multiple feedback loops, multiple interaction, cascade structures, feed-forward loops, interaction between modules, time scale separation. So we need to look at all these aspects when we are writing a manual for a particular phenotype, and then put it in that perspective, right? So only then we understand truly how a input gives rise to a very well defined output, okay, in a context of a system, right? How the systems are going to work. Obviously all these are limited by thermodynamics, limited by rate, the capacity of the system, and it can perturb at many levels in terms of the feedback, and that is why these are complex systems giving rise to very non-linear outputs, right? Which is very difficult to figure out just by the component interaction, right? It is very difficult. Just if I give a map of the network, it's very difficult to say what exactly would be the output for a given input, because you need to look at all these dynamics, correct? So there are, for example, I don't know, in Kerala there is a flowering. A plant flowers once in 12 years. Okay, typically I think in the February, March, in a period of 2 to 3 weeks they flower. Okay, once in 12 years. So how is the system able to do that, right? So it has to keep track of the time, not just in minutes or whatever, decades, right? The hormonal response should happen at the precisely 12-year time scale, okay? But it has to do photosynthesis, it has to do respiration, all that in a daily basis, right? But the flowering should take place once in 12 years, right? So that's how this system has evolved, right? So there is a time scale separations, different components operating at different time scales. So all this is possible in such complex systems, right? So we do look at different network motifs, what kind of outputs they give, and then sort of bring it together to show how a particular system responds. Okay, so I don't know. I don't have time to go through many examples, but typically, what we do is we understand all the knowledge base that exists for a particular system. We bring it down. We put all the constraints, right? Stoichiometry, thermodynamic capacity, because we still are not sure whether we understand completely, right? So we know half knowledge is there, but we cannot sit quiet for the complete knowledge to be available. So we take this unbounded solution space. We put in all the interactions, the topology, the different connectivities. We bring it together. We look at what is the data that is available in terms of stoichiometry, in terms of thermodynamic limits, in terms of the capacity, the rates, so we bring all that together, and then we apply like what Arthur Conan Doyle says in Sherlock Holmes, right? That how often have I said to you that when you have eliminated the impossible, whatever remains, however improbable, must be the truth, right? So systems biology heavily picks on this philosophy that you understand the knowledge, you bring in all the components, you bring in all this network, and then you put all these constraints, right? So whatever is known, it could be omics data constraint, it could be stoichiometric constraints, it could be thermodynamic constraints, right? So we bring in all these time constants, all these constraints that are known. I bring it down, and then in this unbounded space, I look at where I'm landing, right? So because of all these constraints and topology, where do I land, right? And that becomes the probable truth, right? So that is how we try to bring in this input-output concept, and then say how a system is going to operate and how I can build a manual for biological systems, right? So this is how a lot of mathematical modeling, bringing data together, mechanistic details of a feedback, which interactions I need to neglect for a particular phenotype and which I need to account for given the time scales and the interactions. All this becomes very crucial. Model reduction, which interactions to take, right? Visualization, data analysis. So all what the engineers do for a systems approach, right? So we apply to such a system to give rise to very precise output. So I'll just give one example and stop. So for example, genetic network. So this is how yeast controls glucose galactose. So this is a glucose galactose regulon. So we know the network it operates. So I bring in the knowledge base, look at each of these constants, and then build a model on this, and then say, if I'm able to produce and match the experimental data, so you'll see a sensitive response to glucose galactose. So with galactose going on increasing, how it responds. And if I suddenly put glucose, how this comes down, the switch gets switched off. So all this we can do an analysis, and then we do a lot of noise analysis. Why do you have so many feedbacks and all that? And we can show that the system is very robust to noise, both inherent and external noise and internal noise, right? So that the system is able to take care, because you have such a crowded place that the transcriptional factor should come and bind on the DNA, and so there are so many transcriptional factor competing. So this is a highly noisy system. Stochastic noise is very, very high, and we show that this particular design exists. So many things happening here, right? Otherwise, you could have just one transcriptional factor synthesizing. No, the system has evolved so many controls and so many interactions. So why is it, right? So I can now build a manual for this. Say that this interaction is to take care of the noise. This is to make it sensitive response up to a particular galactose concentration. It will not spend energy because it has to optimize its energy also. I will not spend energy beyond this galactose concentration. Only I will express these genes. So all those design elements are inherent in this network, right? And we can actually now show them through feedback sensitivity plots and a noise analysis, sensitivity, right, in terms of input output response, all that analysis we do, okay? And actually can do experimentally measure and show how when you have multiple feedbacks like this, how the noise is so less. Otherwise, you get a very highly a spread out response, but you want a very precise response. So how in different cells you can control the expression system by having these feedbacks, right? So all these kind of questions we can ask now and then actually demonstrate why a particular system exists for a particular thing, right? So in this fashion, we have looked at several systems, and then look at in this context of input output, in context of a systems biology, okay? So let me stop here, and then if anybody has any queries that I can answer.
Yes, we have queries. We very much have queries. So our one of the audience member has a query.
Yeah. Excellent, sir. I'm also from Mumbai IIT and also ICT Mumbai. I stay in Mumbai. Sir, I wanted to know when you presented E. coli.
Yeah.
Account of spores of E. coli is being also been taken. Spores of E. coli.
So yeah. So vegetative cell, okay, is one state. It can go into a spore formation also, right? Bacillus has got a very good decision-making network for when it should be vegetative and when it has to go into a spore formation, right?
Yeah.
And biofilm formation, spore formation. So there is a very elaborate decision-making that the cell does to evaluate when it has to go to a particular phenotype, right? So these are quite well understood. and in a similar context, models can be developed, and then you can understand how the cell under a certain nutritional limitation condition, when does it go into a vegetative state, when it goes into a biofilm state, and when does it go into a complete spore formation. So these kinds of questions we can ask and then say how the cell is taking a decision.
Yeah, thank you, sir. One more, sir, when you said about vegetative propagation regarding some flower and all that. So one of the most important ingredients we get it from vegetative propagation is the medicine, which are phytochemicals.
Correct.
It is being understood from Ayurveda and some other Vedas that in a particular state of a plant, you get the maximum output of the phytochemicals from the plant. Is any work you are doing in that time? What time you have to use the plant?
Yeah. So these principles, what we have is very generic, right? So it can be applied to any system, right? Because it is once you understand, you bring the components together, whether it is a plant system or any other system, you can apply similar principles. And people have done in some plant systems also. But what we have done is in mushrooms, right? So mushroom is somewhere in between, correct? So it is not like it's a fungus, and it does have a fruit body. So we have tried to look at how argothionin, which is one of the phytochemicals which are highly antioxidant. So when does these get accumulated? Under what conditions these phytochemicals get concentrated, right? And what are those conditions that we need to balance in the growth of these mushrooms where these phytochemicals gets concentrated? So some of these things we have done in our lab to optimize certain chemicals to be concentrated, right, in mushroom systems, right? But similar principles can be applied to like what you were suggesting in plant, right? Otherwise you'll have a lot of variability, right? One day you get you have a different phytochemical composition, some other day you take, right, depending on the soil condition, the environment, you'll have a completely different condition.
No, I was talking about the situation of the star and the situation of the sun and all the situation of the moon. As per the changes, the phytochemical changes is being reported in many of the Ayurvedic texts. So phytochemicals are most important chemicals at present required by the drug industry in the world. So
Sure.
if some work you can do for find out a particular plant, a particular phytochemical is available maximum at this time, and those type of phytochemical in their maximum concentration, they only work better. Thank you, sir.
Yeah. So we have optimized the mushroom example I was taking. We have optimized based on the lighting, right? The type of light you put and all that, so that you concentrate a certain phytochemicals in the mushroom. But I know, right? Maybe even the planetary positioning and all that, does affect many of the physiological states. So then, we need to bring that into the context, into the design context. We need to bring those as variables to understand how these metabolism changes in a plant because of positioning of the sun, moon, like what you are indicating, right? So that's a completely different domain. I don't think so anybody has looked at in that perspective.
Is there anybody else in the audience that wants to ask Professor Venkatesh a question? No. Okay. I had a question, sir. The first video that you showed, what was that red box which this was chasing?
So the laser is coming from that end. The immune cell, the optical proteins, right? So the ones which are in the eye. Okay. So in a particular, I think in a frog, in a toad system, in the proteins that are there in the eyes of the toad, so that has been genetically expressed in immune cells, so that they go and reside on the membrane. Okay. So now when you activate a particular laser, right, so they activate the immune response, okay, the signaling pathway inside, and it will start moving towards that light because it is a chemotaxis signal, right? So it will start moving that. So if you change the direction of that, the cell will change, because there is a gradient of a particular PIP3 protein. There is a gradient established inside the cell, and that triggers the motion. Okay, so at one level PIP3 concentration is high, at the other end of the cell the PIP3 concentration is low, and you create that gradient inside the cell, right? So it will start moving in that direction. Okay, and that we have created from a laser light, okay, which activates a certain protein on the membrane, and you can make it move in that direction.
Okay.
Yeah.
That is that your group's work or some source from somewhere?
So it's a collaborative work. So some part of the experiments we did in at our place, but the generation of that protein itself, right? That was done in University of Washington, St. Louis, and the modeling, the system analysis, and all that, and some experiments were done at in our lab. Yeah.
All right. I hope you can visit our institute, and I'm working on the book that you suggested. So let's hope that you can come around and grace our campus one day, sir.
Sure. I'll surely drop in. Unfortunately, I would have come for this symposium as well, but at the right time, I'm sorry, the wrong time. I got this trip to Australia, so that's why I had to do this. Yeah, and
Yeah.
thanks a lot, Murli, for this opportunity. And then I'll
Thank
keep in touch. And I'm looking forward to your book. Yeah, I would want to read it.
Thank you, Professor Venkatesh. Thank you, Professor Venkatesh. Thank you
Yeah.
for taking the time out. Thank you.
Sure. Bye bye. Yeah, I'll be in
Bye-bye.
touch.
Bye-bye. Thank you so much. So, thank you. Danny, can you finish it in 5-10 minutes, or do we take the lunch break? Yeah. I mean, please, I'm asking Danny. One second. Danny needs to take. Yesterday, he was volunteering the other guy, right? So, Danny has to. Yeah, Mera Bharat Mahan. Yeah. No problem, sir. Quickly, huh, Danny? Like Mallu say, quickly. Quickly. Because we have to start the quiz after this. And then there is a talk by Professor Jakin, and then the valedictory.
Okay.
Yeah.
It's working.
Wait.
He'll work.
Yes, I'm happy to see kids at my age.
Okay. Like I told you, we just did simple things here. We have started off with this. It's not like everything has been conclusively done. So the logic is, if you do any molecular docking with this, PGG2 should bind more effectively at which place? Should it be at the cyclooxygenase site or the peroxidase site? It should be at the peroxidase site. And PGI2 also, we took all these molecules and we docked them. Like leukotriene is generated in a very different system. We took thromboxane A2, we took PGI2, we took PGG2, we took PGH2, and we took a bevy of sheep as well as human, different proteins, and tried to check what are the binding energies. And we found that there were lots of discordances, like you could not get, sometimes the substrate is binding better than the product and things like that. We have to do experimentally. No. Yes. Yeah. Yes. So what will happen is, if you see this, the substrate has to go in, right, into the lipid phase. It's released close to the membrane. And because arachidonic acid has got a very high plus value, log P plus 6, it's more soluble in the lipid phase. So it will go in there. But it will go into the active site again. Let's say that's a hydrophobic active site. Once your these compounds are released, they have to come out of the membrane phase and again come all the way around. And the product should not be so favorable. Yes. But we were seeing many instances where that was not so. Don't want to put all that data. Okay. And then we analyze that, see how deep it has gone in the arachidonic acid in the crystal structure. It's like, you see the black, what I have colored, that is the entry channel. And there is a labyrinth there in that protein. I took such a lot of time to mark all these places and to make that channel. It took me probably half a day to do this one image. So you see how it goes in there. And that's, you can see lots of constrictions and all that in those channels. Now, if it converts into a hairpin-like structure because of the cyclooxygenation, like that endoperoxide is generated, becomes, how flexible is it? I don't know. Only dynamics can say. So you got all these other channels also, probably which have no roles in literature. That is the main point in this slide. Okay. And if you can see here, right here, this is the place where it enters. You can see how constricted it is. How much dynamics is there for that to open up? We have inhibitors which are very, very large. Would they be able to enter all the way and go here? This is the tyrosine 385 radical, which is supposed to be involved in attacking arachidonic acid. In the same crystal structure, they had shown that in one place it is not favorable. The CO minus end is entering in. In the other monomer, it's a dimeric enzyme. In the other monomer, they say that there is productive binding. And they are showing that there is a lot of allostery here. Now think of a dimer in which only one of the monomers is active according to their hypothesis. We have so many problems like this because of lack of time. I don't want to go on into all those details, but you see, look at the entry channel. It's so minuscule. And I don't think, logically many inhibitors would be able to enter into this. And how, the entire arachidonic acid is driven in and how it comes out. It's not like it gets sucked in like we suck noodles into the mouth and then spit it out. I don't think how that, I can't imagine how it happens. How is the mechanical force there? So we have all these inhibitors. Next, we docked all these inhibitors. In literature, they say that, this is the second thing in docking. In literature, they say that there are COX-2 specific drugs and COX-1 specific drugs. But we were able to find that the active site is very similar. There is no difference in the arachidonic acid binding site. Most of these inhibitors go well and dock inside. So according to crystal structures of some of these inhibitors, they have made mutants of these different amino acids inside and all that. But there is also literature that tyrosine 385 radical is generated, but there are many other tyrosines. That image I have not put here. We have mapped each monomer as 25 tyrosines. In one paper, they are showing that some tyrosine, which is far away from this tyrosine 385, they mutate that. That also affects the activity of the enzyme. These kind of things, tyrosine 385 radical is supposed to be the most important guy which does all the work. But how are these other tyrosines able to influence? And another crazy thing, which I have not shown, is that there is a surface glycosylation site. It is a N-linked oligosaccharide. There are 4 or 5 of those around the enzyme. It is a glycosylated enzyme. They mutate one of those glycosylation asparagens into something else, and the activity of the enzyme goes up. It is able to work faster. The product turnover is better. How can that be explained? We found in another paper that a similar enzyme, some viral protein, when they mutate the glycosylated that site, the virus or something, I forgot the context, but we have put that in our manuscript. It sops up all the ROS, we would say, according to the murburn perspective, because singlet oxygen is generated here. We have other ROS, we have H2O2, we have superoxide, we have hydroxyl radical. All these things, there will be a dynamics of this making this and all that. So that cannot explain, right? A surface asparagine, you mutate that. Why would the enzyme become more efficient when that is not involved in the enzyme's activity per se? I don't think that could influence the active site pocket. You mutate the active site residues and you show certain things, but this is a data which does not explain, right? Okay. Yes. Yes. So sometimes, you can have better binding in the data that we have. There is better binding of certain molecules in the peroxidase site, not in the cyclooxygenase site. Even that can lead to inhibition. And one thing with all these NSAIDs and other drugs is that you take too much of it, you can also have a heart attack. So how are these non-specificities explained? Why do antioxidants, for example, inhibit cyclooxygenases? Many natural compounds, they inhibit cyclooxygenases. So we have given a perspective. So, no, we have not put. There are a lot of natural compounds which inhibit cyclooxygenase. EGCG, for example, epicallocatechin gallate. So many flavonoids and all. Here you can see that this is a new perspective which we are giving. How does two oxygen molecules have to be consumed there, right? In order to make PGG2 from arachidonic acid. So does the arachidonic acid absolutely need to enter into the membrane? We don't know. According to murburn view, radicals which are generated by the peroxide heme ion can travel and they can also do the catalysis. Because if you think of how you take oil in a pan and you heat it, oxygen, gets into the system and you can get hydroperoxides, right? So there was another paper which we cited, which shows that you can get even stereochemical products because of ROS reactions. Because arachidonic acid is a molecule without any stereo specificity, but when it comes out, there is a lot of stereo specificity. Even radical reactions can do that. So here we have taken two different views, where one is the entry of arachidonic acid inside and the role of tyrosine 380, that is a classical view. So every time, the substrate has to be in the membrane, then it has to go into the active site, then again it has to come out, then that PGG2 has to come all the way around. There are so many problems with this hypothesis. It's not simple. But if we have DROS, which are thought to be wasteful products, are they driving the catalysis? And we think that they absolutely are. I'll give it over to Sir because I don't want to take more time.
Yeah, it's okay. Have you covered all this?
This is our.
Yes. There are lots of observations, data points that don't agree with a purely active site driven thingy. Just showing in one rat system that arachidonic acid enters that place and you can get a crystal structure like that does not mean that it is catalytically, kinetically viable. Why? Because when you put a horrible lot of molecule and try to crystallize something, it can put it in there. It can happen. But does it have a physiological, functional viability? That is what we are interested in. All right. That is a question. Then the aspect that this radical is generated on tyrosine 385 by the tunneling effect by Marcus theory and all that. Why need be? We know that iron can produce radical like superoxide, hydroxyl radical it can generate. It can also give singlet oxygen. Singlet oxygen is very facile, known to be inserting as diatom oxygen. It can give that. So when the when these diffusible reactive species come out and these are bound here and there, they can give some specificities and all that. Because to understand the probability and the scheme of interactions, to have it go in the way the classical cycle goes, it is a very fanatic, very highly fastidious system. The way we propose it, it's very, probability parallel systems, it can happen. So today morning, this was included in the interaction because this is one of the most important systems. And we got a rejection letter today. And you see how it is worded. We are happy that we are getting such nice worded responses. It is
After 2 months,
after 2 months,
there is no review.
nothing. But it's okay. I'm happy.
No, what did they write?
Huh?
What did they? Can you?
Yeah. Thank you for submitting your above reference manuscript to biological chemistry. After careful consideration, we have reached the conclusion that the subject matter of this manuscript is suitable for publication in a more specialized journal. Therefore, we are closing this submission. More specialized journal. What? नहीं नहीं ये सब बहुत कर लिया हमने बहुत देखे हैं ये बहुत this is I love this नहीं 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 this guy this person he did not irritate us so he does not get any bad response. I'll show you some of the responses that I have written. So we are happy to share this with you. Nowadays we don't really strive for publishing in top journals and all that. We just try to put our work ideas out there, even if it is half baked. We have got enough data now in multiple systems to say, to give us our happiness that we are on very solid path. This is an immune system. We know that immunological, if Aileen is listening, when you Aileen Thatcher, Dr. Aileen Thatcher, Professor Aileen Thatcher, she is from US. She is associated with our institute also. So in immunology, what is the thing that elicits something? Now, even that molecule chasing after another molecule, how can they light induce or whatever? The first step is always reactive species. It has to be. That is the chemical way it signals. So proteins are involved. These are also involved. That is what completes the picture. And in immune system, you put aluminum in adjuvants. Why? Raws. Everything. It has to give. So we know we are in decent path now, but the time is now right for you to go and have your lunch. Let us come back quickly for the quiz. And then we have 3:30, we have a talk, and 4 o'clock we have the chief guest inaugurating, the validating things. Yeah, we will